Person was exposed to large amounts of carbon tetrachloride. Light microscopic examination of his liver specimen is shown below. These changes are the result of. Now, this question is testing a fundamental concept in pathology. So, carbon tetrachloride is going to be a toxin, and just like many other toxins that are going to enter our body, they're going to be taken up by the liver in order to be detoxified. So, in the liver, toxins are going to be acted on by the P450 oxidase system. And when carbon tetrachloride is acted on by the P450 oxidase system in the liver, it is going to be converted into a free radical. And the free radical that is going to form is going to be carbon trichloride. So now we have carbon trichloride, and imagine that this is a hepatocyte that I've just drawn. Now, our cells have a sodium potassium pump where we are pumping out sodium and bringing in potassium into the cell. Now, free radicals have the ability to damage the cell membrane. When we convert carbon tetrachloride into this carbon trichloride free radical, it is then going to damage the cell membrane of hepatocytes. So now, because of this damage, this sodium potassium pump is not going to work properly. So now what is going to occur is sodium is basically going to build up within the hepatocyte. And remember that water likes to follow sodium, so now we're going to experience cellular swelling. Now this is important because our organelles, like the rough endoplasmic reticulum, are also going to start up swelling. So imagine that this is the rough endoplasmic reticulum, and the rough endoplasmic reticulum is going to contain ribosomes attached to it, and the ribosomes are going to be important for protein synthesis. And so when the rough endoplasmic reticulum also swells up, these ribosomes are basically going to pop off. And so that is going to ultimately reduce our hepatocytes' ability to do protein synthesis. Now, this is important because one of the other important functions of the liver is to take up fats coming in from our diet and basically repackage them and send it out to other parts of our body that needs to use up those fats for normal cellular processes. And when the hepatocytes send out fats into the blood, it sends them out with a transporter called the apolipoprotein transporter, and the apolipoprotein is going to be a protein. So if we reduce our hepatocytes' ability to do protein synthesis because we've swelled up the cell and the ribosomes have popped off and there's cellular damage, we're ultimately not going to be able to send off those fats with the apolipoprotein. As a result, fats are basically going to accumulate within the hepatocyte. And that is what we're ultimately seeing in this histological slide, where we are seeing these large accumulation of fats within the hepatocyte, as you can see. So these are all basically, these white spaces are fat accumulation within the hepatocytes. Now, all of this occurred because of free radical injury. We've converted carbon tetrachloride to this free radical, and then this free radical started off this cascade of events that led to accumulation of fat within the liver.